It's my absolute pleasure to give a talk today on some of the work that we have been doing over the last 10 plus years. And that has to do with hitchhiking and backpacking. And I'll talk about what's that about, why do we do it, and what are the implications. But before I start, let me give a quick introduction to our work in general. Our work is focused on drug delivery. That's the connecting theme to all the projects which are ongoing in the lab. And fundamentally, the challenge is the biological barriers of the body. When drug delivery is about figuring out how does the drug reach its destination at the right time, at the right dose. And the challenge in accomplishing that is that the body is not designed to carry the drug. It is designed to carry its own metabolic function. So most of the time we find ourselves struggling to overcome these barriers. So over the last 25 years, we have been looking at many barriers in the body. For example, skin, which is a barrier that we have looked at extensively. And if you put a protein formulation on the skin, less than 1% will enter the skin. And that's a challenge if you want to deliver insulin and other proteins transdermally. Intestine is another barrier. It is very difficult to deliver proteins orally. So that happens because the intestine is designed to degrade the proteins and make sure that they don't reach the body. Otherwise, that can be potentially challenged as well. So less than 0.1% of protein typically, if you ingest it, ends up in the body's in the blood circulation. If you go inside the body, the barriers don't go away. They just take a different form. If you deliver nanoparticles in the blood, they all end up in the liver or the spleen because liver is designed to capture the nanoparticles which are freely circulating. And brain, finally, another example of one of the toughest barriers to get into. Not only it is difficult to get into, but whatever gets in is actively pumped out. So when you look at the efficiencies of transport across these barriers, we are looking at 0.1%, 1%, extremely low efficiencies. So over the last 25 years, we have been looking at studying these barriers, including developing mathematical models, developing physical models to study how do drugs transport across these barriers. And more importantly, how do we develop new strategies to overcome these barriers so that we can deliver drugs more effectively? So today I'm going to talk about one barrier in particular, which has to do with nanoparticles which are injected in the body. So the challenge here is the so-called targeting challenge. So when you deliver the drug intravenously, how do you make sure that you deliver the drug more selectively to any particular organ? And the reason targeting is important, and I would say it's more important in the context of either small molecules or nucleic acids. In the case of small molecules, the drug is small. So if you deliver it freely in the body, it will go everywhere in the body because the diffusion is, relatively high for small molecules. So the challenge is not how to get to the target tissue, but how to figure out that it does not get the target that you don't want. How do you avoid off-target toxicity? In the case of nucleic acids, the challenge is that if you freely deliver nucleic acids, they are not stable. So how do you stabilize it? And the solution in both cases has been nanoparticles. In the case of small molecules, if you encapsulate a small molecule in a nanoparticle, you are essentially making a small molecule a large entity, and then you reduce the diffusion. So now you have more control over the background. But now the problem is how do you get these nanoparticles in sufficient quantities in the target tissue? In the case of nucleic acids, when you encapsulate them in lipid nanoparticles or other nanoparticles, you stabilize them. But then the question is, how do you get them to the target tissue? And these nanoparticles can be of any form. They can be lipid nanoparticles, they can be micelles, they can be polymeric nanoparticles. I'm not going to talk so much about any particular type of nanoparticle, but want to talk about uh, the general issues that these nanoparticles face. And most of the nanoparticles that I'm going to be covering today are made up of polymers. So this may be of relevant to those who are coming to the conference from the polymer side, not necessarily the nanoparticle side. So this question about targeting is not new. It's been around in the field for 20, 25 years, and people have figured out two general strategies to address it. The first strategy is you put PEG, polyethylene glycol, on the nanoparticle surface, and that reduces the absorption of serum proteins, and that reduces the recognition by the immune system so that this particle can stay longer. And the longer they circulate in the blood, hopefully, the more targeting that you can get from them. Now, the strategy in principle works, but it's running into more and more challenges, mainly because the body is developing antibodies against PEG. Uh, People have been exposed to PEG in a variety of forms, but more recently, they have been exposed to PEG in the form of lipid nanoparticles used for vaccination. So the body, more and more fraction of the population now has antibodies against PEG, and that potentially defeats the purpose of putting PEG on the surface of the nanoparticles. So the ability to use PEG to extend the circulation moving forward is uncertain. For targeting, people have put molecules on the surface of nanoparticles, either small molecules like folic acid or antibodies or peptides. And one of the issues with the molecule-based targeting is that the for the molecule to really do the function of targeting, it has to come within 1 molecule distance of its binding site. And many times that doesn't happen because the nanoparticles are circulating, they stay in the middle of the stream, they don't even come in contact with the vascular wall. And in the absence of that close contact, the targeting molecule doesn't even see its binding site. So we have been thinking over the last many years, how can we think about an alternate approach to targeting, something that does not necessarily involve the use of molecules or molecule-based targeting? And that's where we started looking at body's own inspiration, so to speak. For everything that we want our nanoparticles to do, there is an example of a cell which does that very well. For example, if we want the nanoparticle to stay in circulation for a long time, look at red blood cells. Each red blood cell stays in circulation for 4 months. It goes past the liver many times. It is not captured. If you're looking for a nanoparticle to bind selectively somewhere in the endothelium, look at what platelets do. They circulate for a long time. They don't bind to anything. But the moment there is damage to the vascular wall, they bind selectively and induce hemostasis. If you're trying to deliver deep into the tumors, look at what macrophages do. They can go centimeters into the tumors, whereas nanoparticles have a tough time getting even tens of microns into the tumors. So the question we have been asking is, how can we leverage this transportation mechanism that the body has built to overcome the barriers and deliver drugs more effectively? And our first solution and thought was, let's just hitch a ride on them. Let's use them as carriers for our drugs so that we can overcome these barriers. So our first example in that category was red blood cells. We wanted to see if we put the nanoparticles on red blood cells, can we extend the circulation and can we benefit from the enhanced circulation of the red blood cell themselves? So what you see in front of you is a red blood cell, an electron micrograph of a red blood cell with a few nanoparticles placed on them. These are polystyrene nanoparticles, model nanoparticles. And one thing to note is that these nanoparticles are physically adsorbed on the red blood cell, not chemically conjugated. And that's important for the reasons will become clear in a few minutes. There are many ways of putting nanoparticles on red blood cells. A lot of the work is published. Happy to answer any questions after the talk. But typically, we can put anywhere between few to a few dozen nanoparticles on red blood cells. And the question now is, does it work? Will the nanoparticles, when they are attached to red blood cells, will they escape the immune system? So that's an interesting question, because if you envision this RBC nanoparticle complex going past the liver is going to see these foreign nanoparticles that it wants to remove as quickly as possible. But they're associated with a surface red blood cell, which it does not want to clear. So who's going to win? So we did the experiment. This was done in a rat model. So if you look at the graph on the right-hand side, it's the particle remaining in circulation as a function of time. And when we inject free nanoparticles, they are cleared very quickly. And that was expected. Polystyrene is really very antigenic. So it is captured very quickly by the liver macrophages. But when we put the same nanoparticle on the red blood cell surface, we see a significant enhancement of circulation. Now we can see the nanoparticles in the blood for up to a day. And if you look at the surface of the nanoparticle, which is away from the red blood cell, if we put PEG on that surface, we can keep the nanoparticles in circulation even for a longer time. We can see them for days in circulation. So that's a dramatic improvement in circulation. It went from a few tens of minutes in the case of free nanoparticles to a few days when we put them on red blood cells. So we were very excited that this strategy does seem to enhance the circulation. But then when we look at the half-life, it's clear that the nanoparticles are disappearing from the circulation. They are not staying there for 4 months, like what the red blood cells do. So the question was, what's going on? One of the concerns was that the nanoparticles somehow are messing up the RBCs. The nanoparticles, by the way, are associating with red blood cells mostly through hydrophobic and electrostatic interactions with the membrane. So if they were disrupting or changing the red blood cell in a way that the whole RBC is now captured by the macrophage, that would not be so good. So we labeled the RBC and the nanoparticles separately, and it turned out that the nanoparticles are cleared from the blood, but RBCs are not. They continue their normal survival in our circulation in the blood. So where are the nanoparticles going? Turns out they go to the lungs. So if you look at the left-hand side graph, where we put doxorubicin nanoparticles in PLGA, and those PLGA nanoparticles were adsorbed on the red blood cells, we see a dramatic improvement in the deposition of these nanoparticles in the lungs. And the reason it's happening is that if you imagine this RBC nanoparticle complex after injecting in the blood coming into the lung capillaries are typically smaller than the red blood cell, actually much smaller. RBC is about 8 microns in diameter. The lung capillaries are 3 micron-ish in diameter. So this RBC is essentially getting extended. It's scraping along the endothelial wall. And the nanoparticle, which is sitting on the RBC surface, is now exposed to essentially a shear or friction between the endothelium and the red blood cell. And it comes off the red blood cell and gets deposited into the endothelium of the lungs. So it's basically a way to transfer nanoparticles from the red blood cell into the endothelium. This transfer does not depend on molecular recognition. There is no targeting ligand on the nanoparticles. It's a purely mechanical, physical way of transferring the nanoparticles from the red blood cells into the endothelium of the lungs. And because it is very physical and mechanical, the efficiency is very high. We can get 50% of the injected dose or up to 50% of the injected dose to show up in the lungs, which is quite remarkable. This is not percent of injected dose per gram, which is how typically is reported. This is actually a significant fraction to 50% of the injected dose that ends up in the lungs. If you don't want to go to the lungs, if you want to go to a different tissue, we can just simply change the injection site. Instead of injecting in the vein, if we inject in an artery, the same mechanism happens in the first tissue, which is downstream from the injection. For example, if you inject in the carotid artery, the nanoparticles experience the same mechanical shear in the capillaries in the brain, and that's where they come off. So if you look at the middle graph, you can see that there is about 25-fold improvement in the nanoparticle deposition in the brain. And if you look at the absolute numbers, we see about 10% of the injected dose to show up in the brain, which is a dramatic improvement over what typically people see with molecule-based or molecular-based targeting to the brain. Same thing happens in kidneys. We see an improved deposition in the kidneys if you inject upstream from the kidney. So we were very excited about this physics or physical and mechanical based targeting of nanoparticles to tissues, because it can provide very high and tissue specific targeting to different tissues. So what can you do with it? So we have delivered a variety of therapies with this. On the left-hand side, these are doxorubicin nanoparticles in PLGA. These are standard PLGA nanoparticles, a few hundred nanometer-ish diameter. We encapsulate doxorubicin and simply adsorb them on the red blood cell. You can see the SEM, the colorized SEM at the top. The blue dots are PLGA nanoparticles. And when we inject them IV, they go to the lungs. And when we test it in the metastasis model, we see a significant improvement in the survival of the mice. So fundamentally, what's happening here is that because a large fraction of the injected dose ends up in the lungs, we can get very high local concentration and we can reduce the off-target effects. And because of that, we can achieve a significant or therapeutically significant concentration of doxorubicin in the lungs in a way that's otherwise not possible. On the right-hand side, you see a graph where we do gene therapy in the brain. So here we are putting viral vectors, AAVs, that can deliver the gene. And same principle. So you can see an SEM. We can see the specks of AAV on the red blood cell. When we adsorb those AAVs on red blood cells and inject in the carotid artery, they come off in the brain and we see increased expression of the gene in the brain. If you look at the top row, there is significantly higher expression in the brain when we hitchhike on red blood cell compared to free AAV. So that was red blood cells. Then we started thinking about what other cells we can hitchhike on. And the next cell that came to mind was a macrophage. And macrophage was interesting because, as I mentioned, macrophage or their precursors monocyte are able to circulate and extravasate and go into the tissues. Red blood cells, while they're effective in depositing nanoparticles into tissues, they never leave the circulation. So if we want to go deep into the tumors, we thought macrophages could be quite interesting. And actually, if you look at a solid tumor, typically a large fraction of the tumor mass is macrophages because they do like to infiltrate into the tumors and they do like to form a substantial mass within the tumor. So that was the motivation, but hitchhiking on macrophages turned out to be quite difficult. In the case of red blood cells, the the RBCs were happy to just act as a platform for nanoparticles. They are not metabolically very active. They are not trying to internalize anything that binds to their surface. So they simply carried our nanoparticles. Macrophages are on the exact opposite end of the spectrum. They are very active. They want to internalize everything that binds to the surface. So we had to figure out a way to keep the nanoparticles on the surface of the macrophage. And that was important because once the particle goes inside the macrophage, it's theirs, it's not yours. So you can't really do a lot of drug delivery if the particles are internalized. So that's where a discovery that we had made many years ago came into the picture. One of my former students, Julie Champion, when she was in my lab, uh, we had found that while macrophages can easily internalize spheres, they cannot internalize disks. So if you look at the micrograph in the middle, the macrophage has easily taken up three large microspheres. Size doesn't really deter them from internalization. But if you look at the rightmost image, the macrophage cannot internalize a disk. And fundamentally, that has to do with the, the dynamics of actin polymerization underneath the membrane. So when a macrophage binds to a surface, actin begins to polymerize, and that drives the membrane around the particle. So if it is trying to go after a sphere, it can slowly expand and engulf the sphere completely. But if it's trying to go after a disk, it cannot coordinate actin polymerization to a level where it can fully engulf the particle. So it tries to internalize, it doesn't stop trying, but it cannot internalize it. So the particle goes for a ride wherever the macrophage goes. It doesn't bother the macrophage. And that's what we call a backpack. So a backpack is a disc-shaped, typically polymeric particle, the way we are using it. And it's about 5 to 8 microns in diameter and about 500 nanometers in thickness. So it's a thin, large polymer disc that sits on the immune cell, goes for a ride wherever the cell goes. The way we make these backpacks is using spin coating. So we have a template, a PDMS template. We can make pillars. The diameter of the pillar governs the diameter of the backpack, so we can control it very easily. We typically don't go much below 3 microns because those can be internalized by the cells. And we typically don't go beyond 10 microns because they can be too big. So our sweet spot is between 5 to 8 microns. And then we spin coat on them to make the backpack. We can try, or we have used different polymers to make the backpacks. PLGA is our typical polymer. We have used hyaluronic acid as well. Any polymer that can hold its shape after the backpack comes off the template can be used to make a polymer. We can also make layers. So we have made different layers of polymers in the backpack. We can modify the surface with different ligands. I'll talk about some of those in a few minutes. Typically, we use antibodies on the surface to bind them to different immune cells. We can load the drug in the backpack while we are making the backpack. So typically we add the drug to the solution that we are spin coating. So we have encapsulated small molecules, proteins, pretty much anything that you can suspend or solubilize in the polymer solution that is spin coated. You can use that to make backpacks. And when the backpacks were spin coated, we can flip and print them, and then we collect backpacks. Typically, we need about 1 million backpacks for a mouse experiment, about 100 million for a large animal study. And for humans, we anticipate needing about 1 billion. And we have done experiments up to large animal studies, so we can make hundreds of millions of these backpacks using this technique. So here is an image, a confocal image of a macrophage carrying 3 backpacks, just to give a feel for the scale of it. And on the left-hand side, this is the point I wanted to highlight, is that one of the key features of the backpacks is that they remain attached to the surface and do not get internalized. If you give them a sphere, no matter how large, macrophages typically internalize it, even like 6 micron sphere, but the backpacks attach without getting internalized, which is very important. So what can you do with it? I'll show you some of the results that we have obtained with backpacks on different cells. So the first cell I'm going to talk about is a monocyte. And so far I talked about macrophage, but I'm switching to monocyte because monocytes are the precursors of macrophages. And what we found in the paper that I cited here is that monocytes are actually much better at infiltrating into tumors compared to macrophages, which is actually quite interesting uh, because that's really what we are counting on, these cells inherently being able to penetrate into the tumors. So the image that you see at the top is a monocyte carrying 2 backpacks. Those red circles are backpacks. And these backpacks are again PLGA. And on their surface, there is CD11B antibody to promote binding of these backpacks to the monocyte. And they also carry interferon gamma. Interferon gamma is a potent stimulator of the macrophage M1-like phenotypes. And the idea here is that when these monocytes carrying backpacks infiltrate into the tumor, they will differentiate into macrophages. And the interferon gamma that is on the surface of these backpacks will continue to drive the M1-like phenotype of the macrophages. Now, I mentioned a while ago that macrophages easily infiltrate into the tumors and they actually form a significant part of the solid tumor. Then, we wonder why are they not doing their job of attacking the tumor? And that's mainly because the tumor microenvironment overrides their ability and turns them into an M2-like phenotype. And the goal here is that to deliver a strong enough dose of interferon gamma to the macrophage so that it continues to maintain its phenotype into an M1-like phenotype, regardless of what the tumor microenvironment is trying to do. And we see an effect of that compared to the injection of free monocytes. On other controls, we see a substantial reduction of tumor growth. Those are the red triangles. And when you look at the survival of these mice, we see an improved survival of the mice. Macrophages are a component of the innate immune system, so they don't have any antigen specificity against the tumor, but by maintaining their phenotype, they are changing the tumor microenvironment, which is stimulating the environment enough to show an anti-tumor effect. We can change the cytokine loaded into the backpack and get the exact opposite effect. Here we try to use the backpack for treating multiple sclerosis, which is an autoimmune indication. And it was interesting because in the world of autoimmune indications, the thought has always been that it's been driven by the adaptive side of the immunity, driven by antigen-specific attack. But the driver for that autoimmune attack is often led by the innate immunity, which causes inflammation in the local tissues. So we wondered whether we can take advantage of the fact that if there is inflammation, monocytes will infiltrate into the tissue. And if we can reduce the inflammation, can we mitigate the autoimmune attack on the tissue? So here we encapsulated interleukin-4 and dexamethasone in the backpack. Both of them are anti-inflammatory agents, and collectively they work highly synergistically. And we put them on the monocytes, again with CD11B antibody to promote adhesion. And we injected them intravenously in mice model of multiple sclerosis, that EAE model. And there are a couple of interesting questions. One certainly was whether the monocytes are able to reduce the inflammation and treat multiple sclerosis, but also a question as to whether we can deliver these backpacks into the brain, because brain, as I mentioned, is a very difficult organ to deliver into. So what we saw here was, compared to controls, we were able to reduce the progression of the disease. So if you look at the green triangles, there was substantial reduction of the disease progression compared to saline control or monocyte control. And that led to improved survival of the mice as well. And when we look at the distribution in the brain and the spinal cord, we see and we see substantial deposition of the backpacks and backpack carrying monocytes into these tissues. When we look at quantitatively, we are seeing about 1.5% of the injected dose of backpacks to end up in these tissues, which is quite significant. And when we look at the presence of inflammatory cells in the spinal cord, we see a significant reduction uh, in the case of backpack carrying monocytes. So again, that indicates that not only the monocytes are able to get into these tissues, but the backpacks are able to control their phenotype and reduce the inflammation. Another application of the same backpack, this was just published a couple of months ago, is in the case of trauma, a traumatic brain injury, TBI. The TBI was interesting because it's a very common condition. It affects about 50 million people worldwide each year. And there aren't really good methods to treat it currently. And not only the trauma part of TBI needs to be treated immediately, but there is also substantial long-term consequences coming from the inflammation in the brain. And that inflammation causes or increases the risk of subsequent autoimmune indications and other brain disorders. So it's very important to reduce the inflammation in the brain after TBI. And we thought that could be an interesting opportunity for macrophages, because when trauma happens, there is inflammation, and that's going to drive the infiltration of macrophages. So if we can put backpacks on these macrophages, can we reduce the inflammation? And this we tested in a pig model, which is a very difficult model to set up. We did that in collaboration with Mass General Hospital. This was just published recently. So if you look at the saline control, you can see the bleeding site, and that's where the trauma is. There is substantial swelling. But when we treat it with backpacks and macrophages, there is a substantial reduction of swelling. In fact, we see about 60% reduction of swelling, which is quite interesting. And when we look deeper into the brain tissue after treatment, we see a significant reduction of inflammatory cells compared to the saline treatment. We see a significant reduction of inflammatory cells, which is really the key objective of a treatment like this. Not only backpacks can be used to treat TBI, but they can also be used to detect TBI. And that is interesting because when TBI is severe, for example, somebody has a fall or an accident and there is bleeding, you know that trauma has happened. But there are many cases where somebody has a fall and there is no immediate sign of trauma, a concussion, even that can be very subtle. And the current way of diagnosing it is that the patients come in and they get an MRI. And MRI, if it shows something, then there is some follow-up treatment. But if there is nothing that shows up in the MRI, patients are asked to go home and then report if they observe or they experience something. Now, MRI is a structural method. It detects changes in the structure of the brain. So trauma might have happened, which is strong enough to produce a biological effect, but not strong enough to produce a structural change. And those trauma go unnoticed. So we thought that's an opportunity again for the backpacks. If we look at the infiltration of macrophages into the brain, even though there may not be a strong enough trauma for the structure to change, there might be enough of a change for enough of a trauma from the biological perspective for the macrophages to sense it and infiltrate into the brain. So on the left-hand side, you see some images of backpacks, green on macrophages. And here we put gadolinium in the backpacks as a contrast agent. And because it's gadolinium, we had to change the backpack because gadolinium requires contact with water. That means water has to penetrate into the backpack to get close to gadolinium. So for that, we use hydrogels as a material. So these are hyaluronic acid backpacks. And on the right-hand side is the data in pigs, also in collaboration, this time with Boston Children's Hospital. And the images may be difficult to interpret if you're not used to seeing brain MRIs. But what we see is that compared to a standard MRI, which also uses gadolinium, we don't see an effect of the trauma. But when we look at the contrast generated by the gadolinium containing backpacks on macrophages, we are able to see trauma in pigs, which can be below detection by the conventional MRI method. So in all these cases so far that I talked about, monocytes, macrophages, cancer, multiple sclerosis, TBI, we always had something in the backpack, whether it's a small molecule or a cytokine or an imaging agent. But as we were doing this experiment, we started noticing that even without anything in the backpack, it's having an interesting effect on the cell. So the image here in front of you is a T cell, purple, that is attached to a backpack that's in pink, just so that you get an idea for the scale of it. And what seems to be happening is that if you look at the world of interactions with cell membranes, if you have one molecule, let's say one protein or one antibody binding to one receptor on the cell, that interaction is one molecule wide, so like 10 nanometer, let's say. If you take a classical nanoparticle and put like 100 nanometer nanoparticle and put antibodies around it, you may be engaging a few hundred molecules on the surface of the cell. But if you have a backpack and if you have an antibody on the surface of the backpack, when that binds to a cell, it is engaging hundreds of thousands of molecules or receptors on the cell. So that level of massive interaction with the membrane is causing effects which are substantial enough that that alone is driving interesting biology. For example, here we put a PLGA backpack which had CD45 on the surface to promote adhesion to a cell and NKP30 to activate the NK cell. So the image at the bottom left shows an NK cell, a natural killer cell with two backpacks in red. And what's happening is that if you look at the bottom figure, it is activating the cell in a size-dependent manner. As the size goes from or increases from 4 microns to 10 microns, the activation, as measured by the release of interferon gamma, systematically increases. And there is nothing, there is no drug in the backpack. There is no molecule, there is no cytokine. It's just the PLGA backpack that is binding to the NK cell. And if you take the same backpack and kind of divide it into a number of small nanoparticles and put all those nanoparticles around the cell, that does not lead to the same activation as shown in the top graph there. So it's really the act of a single large surface that is engaging with the cell causes the activation of the cell. And when we inject these cells carrying backpacks, NK cells, into mice, we see that they are better at attacking the cancer and they reduce the metastasis of, in this case, melanoma tumors in the lungs. Same thing also happens with neutrophils. So here you see a neutrophil attached to two backpacks in the top panel. And again, there is no drug. It just an antibody to bind the backpack to the surface, CD11B, and that activates the neutrophil as measured by the release of myeloperoxidase. And when we inject these activated or backpack activated neutrophils in mice carrying melanoma tumor, we see a reduction of tumor growth, increased survival, especially when we co-administer checkpoint inhibitors. And those mice that survive at the end of the experiment, if we re-challenge them, they reject the tumor. And this is interesting at multiple levels. One is that we are seeing an anti-tumor activity without any drug, so to speak. PLGA backpack is the drug. And the activity is mediated through neutrophil. Neutrophil again is does not have antigen specificity, so it is activating the tumor microenvironment, and that's where the activity is coming from. And neutrophils themselves are really short-lived. In the world of cell therapies, neutrophils are not really looked at because they live for about a couple of days. So why would you inject a cell that won't live without 2 days? That's why they haven't received any attention. But what we are seeing is that even though they live for a short period of time, in those 2 days, they are producing an effect in the tumor, which can produce a long-term effect on the outcome. So that's really interesting. Same thing also happens in the case of glioblastoma. It's the same backpack on neutrophil. And when we inject it intravenously and look at the efficacy against the glioblastoma model, which is very difficult to treat, the graph in the middle is the subcutaneous model of glioblastoma, and the figures on the right are the orthotopic model in the brain. And you can see that there is increased efficacy in both cases by neutrophil attached to backpack. Again, no drug attached to, and no drug encapsulated in the backpack. It's just the PLJ backpack that's activating the neutrophil. And my final example, this is something we are actively working on, is the T cell. So here you see a T cell attached to a backpack. The green blob is the T cell, the red is the backpack. And on the right-hand side, you see the SEM. So the cell looks normal like any other T cell. It's moving around like a normal T cell. But when we look under the hood, we see significant activation. So activation indicated by the interferon gamma secretion compared to cells alone, we see a significant improvement in the activation. And when we inject these activated T cells in the mouse model, we see reduced tumor growth, and that also leads to improved tumor survival. And just to make a point about the contact between the backpack and the cell, when we look at the SEMs, and in some cases, we can kind of remove the backpacks after the cells are fixed, and you can see the large footprint that the backpack has produced on the cell. It is that engagement of the backpack with the cell for that large area, which is what driving this interaction. So taking a step back, looking at these results, this is one of the reasons why we are excited about polymeric backpacks as a new therapeutic. The activity within the human body, whether it's in health or disease, is controlled by the immune cells. I showed you a few examples: macrophages, neutrophil, NK cell, T cells, monocyte, B cells. All of these have very interesting properties. For example, macrophages are very versatile, but they don't hold their phenotype. Neutrophils are the first to respond to inflammation, but they don't live for a long time. T cells are extremely efficient in killing cells, but they get fatigued. That's why they work in liquid tumors, not solid. So these cells are inherent to essentially health and disease outcomes. And by being able to use backpacks on them, we believe that we can enhance their strengths and mitigate their weaknesses. And by doing that, we can get better functionalities from these cells. So as we are going towards cells as therapeutics, the world of therapeutics has evolved from small molecules to proteins to nucleic acid going to cells. So as we look at cells as the next generation therapeutics, the ability of the backpacks to control the cell's phenotype and behavior opens up new opportunities in cell therapies. And even taking one further step back, one of the exciting things about backpacks is that the world of cell polymer interactions has always been divided between the two worlds. There's a world of drug delivery where the polymer particle is small and then it binds to a cell, gets internalized, and does its thing. Or the world of tissue engineering where the polymer substrate is large and the cell binds to it and spreads on it. And the backpacks are right in the middle. Backpacks are large enough to make the cell think that it's a substrate so that it can bind to it, it can grow on it, and it can attach to it. At the same time, backpacks are small enough so that the cell can take it with them wherever they go. So in a way, that's a mobile support platform which can provide mechanical support to the cell like a substrate. It can provide a chemical support to the cell. It can provide biological support to the cell. It can provide a therapeutic support to the cell like drug delivery carrier. So they really blend ideal properties from both worlds. So we have put backpacks on all kinds of cells. I showed you some examples of neutrophils, monocytes, macrophages, red blood cells, B cells, NK cell, T cells. And in each of these cases, they really provide interesting opportunities, and collectively they can take on multiple diseases from cancer to autoimmune disease to neurological conditions, trauma, wound healing, etc. So that's one of the reasons also why we think this is a very interesting platform for therapeutic applications. So let me stop here and answer any questions. I want to thank my students whose work I got to present here today. Very grateful to them for all their hard work and dedication. A lot of collaborators who have helped this research along the way, some of whom I mentioned along the way. And on the right-hand side is a cartoon of a cell with a backpack. So if there's sort of one image that you want to think about, in terms of backpacks on cells, this could be it. Thanks to funding sources. Thanks again, Sergey, for the invitation. And if there is time, I'll be happy to answer any questions.
Over to you, Twana.